Good evening, sir. This is Prabha Viswas. I welcome you to this short presentation. My gene of interest is CRISPR-Cas edited CCR5 delta 32 of Homo sapiens. It is located in short arm of chromosome 3. And delta 32 implies the mutation of 32 base pairs. This mutation can be spontaneous or in the lab, and spontaneous mutations occurs in the Northern European population in about 15 to 20% cases, also in Asian population, but with a lesser degree, about 5% cases. And this CCR5 gene encodes a chemokine receptor. This is a G protein coupled receptor in T cells and macrophages for immune protection. Natural deletion or point mutation in homozygous state confers protection against the HIV and flavivirus, and this is due to the mutated gene produces shorter receptors, so HIV and other flavivirus can't bind with this receptor, and here lies an opportunity for the HIV patients. And different researchers have found that stem cell transplantation of this mutated gene produces a cure for the HIV patients as well. It also improves the synaptic plasticity and reduces inflammatory responses. Aim of this study is to cloning this mutated gene in the stable mammalian expression vector. This is pcDNA 3.1 and its validation and transmission in the HEC293 cells. pcDNA 3.1 has been selected because it is compatible. It is a large vector. It can accommodate up to 5 kilo base pairs, and it has a strong CMB promoter zone. And it has multiple cloning sites, including different restriction endonucleases like ASC1, HIND3, EcoR1. And it has different antibiotic resistance genes like neomycin and ampicillin. Besides, HEK293 cells are the immortal cells. They are easy to grow, easy to be transfected, as well as these are suitable cells for the receptor protein analysis. With the primer designing of the mutated gene and its insertion into the expression vector, forward primer and reverse primer are designed with the help of the DNA polymerase in presence of nucleotides, and buffer PCR products are produced. Subsequently, these PCR products and plasmids are digested with the help of endonuclease enzymes. This is ASC1 and HIND3, and these would be validated with the help of 1.1% agarose gel, which would cleave the base pair positions of the PCR products and plasmid DNA, and this DNA would be ligated by the T4 DNA ligases. The next step would be transformation of the bacteria to amplify the expression vector and selection of colonies containing the expression vectors. For this, one-shot top 10 chemically competent E. coli would be selected and would be exponentially grown. Later centrifugation would be done to extract the pellet, and following subsequent procedure, the ligated plasmid would be added and it would be incubated, and later part it would be grown in a media containing the neomycin for selective colony selection. Next step is QC validation to ensure that correct expression vector has been generated. The colony would be grown for 1 day, and the next day colony would be selected and the plasmid DNA would be isolated and purified with the help of Thermo Fisher GeneJet plasmid mini rep kit and spin column. In the next part, it would be validated using 1.1% agarose gel to find out the base pair position of the clipped plasmid DNA and PCR products, and the nucleotide sequencing would be done by the Sanger sequence. And the final step is to transfect the HEC293 cells with the expression vectors. At first, the efficient HEC293 cells would be selected by the neomycin kill curve. Then cationic lipofection of these cells would be done by the lipofectamine 2000. And neomycin selection to determine the cells would be done to identify that these cells had been incorporated with the expression vector. In the final part, the visualizations of this mutated gene, that means the CCR5 delta 32 protein, would be visualized with the help of anti-flag antibodies to the DTK tags. And thank you so much for your patience hearing.